So, continuing with part C of chapter 11, do a little backtrack retrograde. So, neurotransmission can be either an excitatory or inhibitory potentials. And I want to show you the difference between the two. So, excitatory synapses, this is when the binding of a neurotransmitter will allow simultaneous flow of sodium and potassium in opposite directions. As a result of that, we ended up getting a net graded potential depolarization where you would see a positive deflection as shown here on this image here. So in this case, here's a stimulus, sodium potassium channels open up, more sodium enters into the cell than potassium going out, and as a result of that, we get a depolarization becoming less negative. In this case, it shows you that it didn't reach threshold, and obviously the neurotransmitter is quickly neutralized by the three processes that we talked about earlier, and it reestablishes the resting membrane potential, and no action potential was achieved. Now, in the case of inhibitory, in this case, when the neurotransmitter binds, it makes the postsynaptic membrane more permeable to potassium alone or chloride ion. If potassium channels open, it moves out of the cell, resulting in more negative, in other words, hyperpolarization. If chloride ions open up, chloride ions move from extracellularly to intracellularly into the cell, which makes the cell again more negative, so hyperpolarization. So in this case, we don't see a positive deflection like we saw in the excitatory. We see a negative deflection moving away from threshold. As a result of this, this inhibits any action potential to occur. So that's what you would see here. So summation of by the postsynaptic neurons. So we can add, several stimuli to have a greater action potential, create a greater depolarization or hyperpolarization, depending upon the needs. And there are two types. There's temporal and spatial summation. The temporal summation, one or more presynaptic neuron transmit impulses in rapid fire order, so very quickly. Spatial summation is when you have many neurons stimulating one structure simultaneously. So here what we can see with regards to, there's no summation in this case. So here, E1, stimulus, didn't reach threshold, and reestablishes. No action potential occurred. So by the time it reached the resting membrane potential, we gave the same stimulus again, boom, the amplitude of the depolarization is going to be exactly the same. However, if the stimulus didn't wait until it reestablished the resting membrane potential and occurred shortly after the depolarization and before it became totally back to the resting membrane potential, these stimuli and depolarization is added to even more. So once it hit the threshold, action potential occurs, we have the depolarization occurs, and then the repolarization, hyperpolarization, and then reestablished. This also occurs with the inhibitory as well. This is spatial summation. Essentially, the result is the same, but in this case, we have two different stimuli stimulating one nerve simultaneously. So it is an additive effect. And again, it reaches threshold where action potential is generated, and the stimulus goes all the way. In this case, if we have an excitatory stimulating at the same time as an inhibitory, they are working in opposite directions. So whichever one dominates is the one where you're going to have the overall effect. So if the stimulus of E1, excitatory, is stronger than the stimulus of the inhibitory, then you're going to have a net positive deflection, but not fully because you had the opposing actions of the inhibitory. If the inhibitory and the excitatory stimuli were exactly the same but opposite, there would end up essentially both of them cancel each other out, and you would have no essentially or very little deflection, whether it's going to be in the positive depolarization or hyperpolarization phase. So in your textbook, you have this table, and it shows you essentially table 11.2, and it shows you here graded potentials and action potentials. With regards to the graded potentials, the initial effect of the stimulus. So here, if we have an excitatory, we have both sodium and potassium gates are open by a ligand neurotransmitter, but more sodium enters than potassium exits, and you get depolarization. So you have a positive deflection. Inhibitory will open either potassium gates or chloride gates. In this case, you are creating the inner membrane to become more negative. In other words, hyperpolarize, and you get a negative deflection. You become more negative. Action potentials is plus 30 to plus 50 millivolts, and in this case, it is the sodium channels that causes the depolarization. With regards to repolarization, it is the potassium channels are open and not the sodium. Next, neurotransmitters. So as I stated in our study guide, I will just have you responsible for just a couple of the neurotransmitters, since there are many of them. And they're classified by chemical and functional aspects. So the first one, as we learned already in the muscles, is acetylcholine. And acetylcholine, we're going to be talking about this more when we deal with the autonomic nervous system. We discussed that the neurotransmitter is the one responsible for somatic impulses, namely stimulating skeletal muscle. And the other is going to be part of the biogenic amines, the catecholamine. Essentially, norepinephrine is the other neurotransmitter that you are going to be responsible for. So now the next is neural integration. So neural integration is how neurons function together in groups. And the groups can contribute to a broader neural functions. And there are billions of neurons in the central nervous system, and they must have integration so that the individual parts fuse to make a smoothly operating whole. So we have this neuronal pool, which are functional groups of neurons that integrate incoming information received from receptors or other neuron pools, and they forward the processed information to other destinations. So it's kind of like a relay station. So with regards to these discharge and facilitated zones, as far as this is concerned, I want to show you how simple neuronal pools and how they are. So here we have a presynaptic neuron that branches, and we have the facilitated zone here, the discharge zone here, and another facilitated zone here. So you can see how one can distribute to different components. So serial processing is inputs travel along one pathway to a specific destination. So one neuron stimulates the next one, which stimulates the next one, and so forth. And systems work in an all or none manner to produce specific anticipated responses. And the best example of serial processing is a spinal reflex. So reflexes, what are these? Reflexes are essentially, let me show you the picture here. We have a stimulus here. Let's say this is a sensory stimulus, and there we have the receptor here, and then the impulse goes from the periphery into the central nervous system via the spinal cord by way of the dorsal root ganglia, dorsal root. And then this communicates with an interneuron, the sensory neuron, the integrates, and then this processes the information, and then it will send out an impulse, in this case, exiting out of the spinal cord to an effector organ, namely, let's say, a skeletal muscle. So, and this comes out via the ventral root. So you don't see it here, but this is a culmination of a group of these sensory neurons here called the dorsal root and motor neurons here, which would be the ventral root. Parallel processing. Inputs travels along several pathways. Different parts of circuitry deal simultaneously with the information. So we have different circuits on how these nerves are connected with each other. So these circuits are four types. We have diverging, we have converging, reverberating, and parallel after discharge. So this is an example of diverging circuit. So in this case, you have one input and it branches accumulatively here to many outputs. So here, a single neuron in the brain can activate 100 or more motor neurons in the spinal cord and thousands of skeletal muscle fibers. So this is a diverging circuit. The second one is a converging circuit, essentially the opposite, where different sensory stimuli can all elicit the same memory. So here we have different inputs coming to a central neuron here, and then this neuron gives the appropriate output. Reverberating, as you can see here, we have an input here going and circling, and then it forms this oscillating circuit. And this is involved in breathing, sleep-wake cycles, repetitive motor activities, such as walking. So this is something that assists and makes our nervous system more efficient. The last one here is parallel after discharge circuits. So this may be involved in exacting mental processes such as mathematical calculations and so forth. So this is called a parallel circuit. So here, the nervous system developmental aspects, some things that you need to know, that the nervous system originates from, it's called a neural tube, and these neural crests are formed from the most outer germ layer called the ectoderm. Some other developmental aspects of neurons. About 2/3 of neurons die before birth. If axons do not form a synapse with their target, they are triggered to undergo apoptosis, which is called programmed cell death. Many other cells also undergo apoptosis during developmental stage. Neurons are amitotic after birth. In other words, these will not undergo mitosis. However, there are a few special neuronal populations that will continue to divide and so forth, like the olfactory neurons and the hippocampus. And this ends the video segment of chapter 11. Thank you.